When I was 8, I climbed into an aluminum rowboat with the elderly director of a summer camp north of Toronto. He rowed a quarter-mile out into the shallow bay, and we spent the next 2 hours fishing. It was a calm summer evening, and the water was like glass. It was my first time in a small boat, and floating on this vast, faintly undulating expanse of dark water was exhilarating. I wondered what creatures lurked below, and this stoked my excitement whenever the sudden jerk of my primitive fishing pole, a stripped sapling with a line and hook, signaled that a fish had struck the bait. I caught 16 fish that day. Some we released. Several others, larger bass and perch, we kept for breakfast the next morning. Mr. Nelson did all the dirty work, baiting the barbed hooks with writhing earthworms, twisting the wire out of the fish's lips, plunging his knife into their skulls to kill them. His face contorted strangely as he performed these tasks, and I wondered if he was feeling revulsion or if he was merely lost in concentration. I have some fond memories of that experience. But, as a sensitive boy with a soft spot for animals, I was disturbed by a lot of what went on in that rowboat. I worried privately about the worms. I fretted that the fish felt pain as the stubborn hook was extracted from their bony, staring faces. Maybe one of the keepers survived the knife and was dying slowly in the wire basket dangling over the side. But the kind man sitting at the bow didn't seem to think there was anything wrong, so I rationalized that it must be okay. And the taste of fresh fish at breakfast the next morning left only vague remnants of misgivings from the previous evening. That was not my only childhood encounter with fish that raised conflicting emotions about our cold-blooded cousin's place in our moral calculus. In 4th grade, I was one of a few kids recruited to move some supplies from our classroom to a neighboring room at my elementary school in Toronto. Among the items was a glass fishbowl containing a lone goldfish. The vessel was 3/4 full of water, and quite heavy. Concerned that the fish not be placed in the hands of someone who might care less than I did, I volunteered to transport the bowl to its destination, a counter next to the sink in the adjoining room. How ironic. I firmly held the bowl in my child's hands and methodically walked out the door, down the hall, and into the new room. As I gingerly approached the counter, the bowl slipped from my grasp and smashed on the hard floor. It was a moment of horror that played out in slow motion. Shards of glass splintered and water sloshed across the floor. I stood there stunned. Someone with more wits than I grabbed a mop and moved the glass and water to one side, then 4 of us began to scour the floor for the fish. A minute went by with no sign of the creature. It was like a bad dream. It seemed as if she had experienced goldfish rapture and risen up to the fishy heavens. Finally, someone found her. She had bounced behind a radiator and ended up on the inside lip, 2 inches above the floor and completely out of view. She was still alive, gawping meekly. She was quickly plopped into a beaker of tap water. I believe that fish survived. Though the goldfish incident left a deep impression on me, as evidenced by my vivid recollection of it 4 decades. Later, I was not moved to a new empathy for fish. Admittedly, I never took a shine to fishing. What little enthusiasm remained after the outing with Mr. Nelson soon faded when it came time to bait and extract my own hooks. But I made no connection between the perch and bass I unceremoniously hauled up from Sturgeon Bay, or the hapless little goldfish I dropped at Edithvale Elementary School, and the anonymous fish who ended up in the filet-o-fish sandwiches I enjoyed on family trips to the local McDonald's. That was the late 60s, and already McDonald's was boasting over 1 billion served. They could as soon have been referring to fish or chickens as to customers. But like other members of my culture, I was blissfully removed from the once living, breathing creatures who ended up in my lunch. It was not until I took an ichthyology course in the final year of my undergraduate biology degree 12 years later that I began to seriously question my relationship to animals, including fish. I was as captivated by fish's diverse anatomy and adaptations as I was disturbed by the parade of inert, once living bodies we were given to classify using dissecting microscopes and taxonomic keys. The class made a midterm visit to the Royal Ontario Museum, where we met one of Canada's foremost ichthyologists for a private tour of the museum's fish collection. At one point he unlocked and raised the lid of a large wooden case to reveal an enormous lake trout floating in an oily preservative. The fish, weighing a record 103 pounds, had been caught on Lake Athabasca in 1962. Her size and plumpness were attributed to a hormone imbalance that had rendered her sterile. Energy that would normally have been spent on the profligate task of egg production was instead assigned to body mass. I felt for that fish. Like most we encounter, she had no name and her life was a mystery. I felt like she deserved a more dignified existence than entombment in a wooden case. To me, it would have been better had she been eaten, her tissues recycling back into the food chain, than to float for decades in darkness, polluted by chemicals. Legions of books have been written about fish, their diversity, their ecology, their fecundity, their survival strategies. And several bookshelves can be filled with books and magazines about how to catch fish. To date, however, no book has been written on behalf of fish. I'm not referring to the conservationist message that decries the plight of endangered species or the overexploitation of fish stocks. My book aims to give voice to fish in a way that hasn't been possible in the past, thanks to breakthroughs in ethology, sociobiology, neurobiology, and ecology. We can now better understand what the world looks like to fish, how they perceive, feel, and experience the world. In researching this book, I have sought to sprinkle the science with stories of people's encounters with fishes, and I will be sharing some of these as we go along. Anecdotes carry little credibility with scientists, but they provide insight into what animals may be capable of that science has yet to explore, and they can inspire deeper reflection on the human-animal relationship. What this book explores is a simple possibility with a profound implication. The simple possibility is that fishes are individual beings whose lives have intrinsic value, that is, value to themselves quite apart from any utilitarian value they might have to us, for example as a source of profit or of entertainment. The profound implication is that this would qualify them for inclusion in our circle of moral concern. Why bother? There are two main reasons. First, fishes are, collectively, the most exploited category of vertebrate animals on Earth. Second, the science of fish. Sentience and cognition has advanced to a point that it may be time for a paradigm shift in how we think about and treat fishes. Just how exploited are they? One author, Alison Mood, has estimated, based on analysis of Food and Agriculture Organization fisheries capture statistics for the period 1999. 2007, that the number of fishes killed each year by humans is between 1 and 2.7 trillion. To get a handle on the magnitude of 1 trillion fishes, if the average length of each caught fish is that of a dollar bill and we line them up end to end, they would stretch to the sun and back, a round trip of 186 million miles, with a couple hundred billion fishes to spare. Mood's estimate is exceptional because the human toll on fishes is rarely presented as a number of individuals. To wit, the Food and Agriculture Organization itself estimated the 2011 commercial fisheries catch to be 100 million tons. Fish biologists Stephen Cook and Ian Cowkes, among the few to enumerate individual deaths, estimated in 2004 that some 47 billion fishes were being landed recreationally worldwide every year, of which some 36% were killed and the remainder returned to the water. If we apply their estimated average weight per fish to a commercial catch of 100 million tons, we arrive at an estimate of 157 billion individual fishes. One study reports that official statistics on global fish catches have been underestimated by more than half over the last 60 years, due to often neglected small-scale fisheries, illegal and other problematic fisheries, and discarded bycatch. However you slice it, it's a lot of fishes, and they do not die nicely. The leading causes of death for commercially caught fishes are asphyxiation by removal from the water, decompression from the pressure change of being brought to the surface, crushing beneath the weight of thousands of others hoisted aboard in massive nets, and evisceration once landed. Regardless of which estimate you take, dizzying numbers like these tend to mask the fact that each fish is a unique individual, not just with a biology, but with a biography. Just as each sunfish, whale shark, manta ray, and leopard grouper has a distinctive pattern from which you can recognize individuals on the outside, each has a one-of-a-kind life on the inside, too. And therein lies the locus of change in human-fish relations. It is a fact of biology that every fish, like the proverbial grain of sand, is one of a kind. But unlike grains of sand, fishes are living beings. This is no trivial distinction. When we come to understand fishes as conscious individuals, we may cultivate a new relationship to them. In the immortal words of an unknown poet, nothing has changed except my attitude, so everything has changed. Part 1. The Misunderstood Fish. We shall not cease from exploration, and the end of all our exploring will be to arrive where we started and know the place for the first time. T.S. Eliot. What we casually refer to as fish is in fact a collection of animals of fabulous diversity. According to FishBase, the largest and most often consulted online database on fishes, 33,249 species, in 564 families and 64 orders, had been described as of January 2016. That's more than the combined total of all mammals, birds, reptiles, and amphibians. When we refer to fish, we are referring to 60% of all the known species on Earth with backbones. Almost all modern fishes are members of one of two major groups, bony fishes and cartilaginous fishes. Bony fishes, scientifically termed teleosts, make up the great majority of fishes today, numbering about 31,800 species, including such familiar ones as salmons, herrings, bases, tunas, eels, flounders, goldfishes, carps, pikes, and minnows. Cartilaginous fishes, or chondrichthyans, number about 1,300 species, including sharks, rays, skates, and chimeras. Members of both groups have all 10 body systems of the land-dwelling vertebrates: skeletal, muscular, nervous, cardiovascular, respiratory, sensory, digestive, reproductive, endocrine, and excretory. A third distinct group of fishes is the jawless fishes, or agnathans, a small division of about 115 species comprising lampreys and hagfishes. We conveniently classify animals with backbones into 5 groups: fishes, amphibians, reptiles, birds, and mammals. This is misleading because it fails to represent the profound distinctions among fishes. The bony fishes are at least as evolutionarily distinct from the cartilaginous fishes as mammals are from birds. A tuna is actually more closely related to a human than to a shark, and the coelacanth, a living fossil first discovered in 1937, sprouted closer to us than to a tuna on the tree of life. So there are at least 6 major vertebrate groups if one counts the cartilaginous fishes. The illusion of relatedness among all fishes is partly attributable to the constraints of evolving to move efficiently in water. The density of water is about 800 times greater than that of air, so aquatic living has, in vertebrates, tended to favor streamlined shapes, muscular bodies, and flattened appendages that generate forward propulsion while minimizing drag. Living in a denser medium also greatly reduces the pull of gravity. The buoyant effect of water frees aquatic organisms from the ravages of weight on terrestrial creatures. Thus, the largest animals, the whales, live in water, not on land. These factors also help explain the small relative brain size of most fishes, which has been used against them in our cerebrocentric view of other life forms. Fishes benefit from having large, powerful muscles to propel them through water, which is more resistant than air, and living in a practically weightless environment means there is no premium on limiting body size relative to brain size. In any event, brain size is only marginally meaningful in terms of cognitive advancement. As the author Cy Montgomery notes in an essay on octopus minds, it is well known in electronics that anything can be miniaturized. A small squid can learn mazes faster than dogs do, and a small goby fish can memorize in one trial the topography of a tide pool by swimming over it at high tide, a feat few, if any, humans could achieve. The earliest fish-like creatures arose in the Cambrian period, some 530 million years ago. They were small and not very exciting. The big breakthrough in the evolution of fishes was the appearance of jaws about 90 million years later in the Silurian period. Jaws allowed these pioneer vertebrates to grab and break up food items and to expand their heads to powerfully suck in prey, which greatly extended the available dinner menu. We might also think of jaws as nature's first Swiss Army knife, for they come with other functions, including manipulating objects, digging holes, carrying material to build nests, transporting and protecting young, transmitting sounds, and communicating. Having jaws set the stage for an explosion of Piscine life during the Devonian period, also known as the Age of Fishes, including the first superpredators. Most of the Devonian fishes were placoderms, having heavy, bony armor over the head end and a cartilaginous skeleton. The largest placoderms were formidable. Some species of Dunkleistus and Titanichthes measured well over 30 feet. They had no teeth, but could shear and crush, with 2 pairs of sharp bony plates forming the jaws. Their fossils are often found with boluses of semi-digested fish bones, suggesting that they regurgitated these in the manner of modern owls. Although they all went out with the Devonian and have been gone for over 300 million years, nature was kind to the placoderms in preserving some specimens so delicately that paleontologists have been able to deduce some intriguing facets of their lives. One particularly revealing find, from the Gogo fossil sites of Western Australia, is Meterpiscis attenboroughii. Named for the iconic British nature documentary presenter David Attenborough, who waxed enthusiastic over this species in his 1979 documentary series Life on Earth. This perfectly preserved 3D specimen allows careful peeling away of layers to reveal the insides of the fish. And what should show up there but a well-developed baby Meterpiscis attenboroughii attached to its mother by an umbilical cord. This discovery rocked the evolutionary boat by setting back the origins of internal fertilization by 200 million years. It also eroticized the lives of early fishes. As far as we know, there is only one way to achieve internal fertilization: sex with an intermittent organ. So it appears that fishes were the first to enjoy the fun kind of sex. About this discovery and John Long, the Australian paleontologist who brought it to light, Attenborough expressed ambivalence during a public lecture. This is the first known example of any vertebrate copulating in the history of life. And he names it after me. Sex notwithstanding, the bony fishes, which arose about the same time as the placoderms, had a brighter future. Although they suffered major losses during the third great extinction that closed out the Permian period, they steadily diversified over the next 150 million years of the Triassic, Jurassic, and Cretaceous periods. Then, about 100 million years ago, they truly began to flourish. From that time to today, the number of known families of bony fishes has more than quintupled. Fossil records do not divulge their secrets willingly, however, so there may be many earlier fish families still hidden in the rocks. Like their bony counterparts, the cartilaginous fishes also steadily recovered from the Permian setback, albeit without the explosive diversification of later times. As far as we know, there are more kinds of sharks and rays today than at any point in history. And we're beginning to discover that their real lives belie their pugnacious reputation. Diverse and versatile. Because their lives are more difficult to observe than those of most terrestrial animals, fishes are not easily fathomed. According to the National Oceanic and Atmospheric Administration, less than 5% of the world's oceans have been explored. The deep sea is the largest habitat on Earth, and most of the animals on this planet live there. A 7-month survey using echo soundings of the mesopelagic zone, published in early 2014, concluded that there are between 10 and 30 times more fishes living there than was previously thought. And why not? You might have encountered the popular notion that living at great depths is a terrible hardship for the creatures there. It's a shallow idea, for surely deep-sea creatures are no more inconvenienced by the enormous pressure of the overlying ocean than we are by the approximately 10 tons per square meter pressure of the atmosphere above us. As the ocean ecologist Tony Kozlow explains in his book The Silent Deep, water is relatively incompressible, so deep-sea pressures have less impact than we usually think, because pressure from within the organism is about the same as that on the outside. Technology is just beginning to afford us a glimpse of the ocean depths, but even in reachable habitats, many species remain undiscovered. Between 1997 and 2007, 279 new species of fishes were found in Asia's Mekong River basin alone. The year 2011 saw the discovery of 4 shark species. Given the current rate, experts predict the total count of all fishes will level off at around 35,000. With the advance of techniques for distinguishing species at the genetic level, I think it could be many thousands more than that. When I studied bats as a graduate student in the late 1980s, 800 species had been identified. Today, the count has ballooned to 1,300. From diversity springs variety, and from the rich variety of fishdom spring some noteworthy superlatives and bizarre life patterns. Thesmolestfish, indeed, the smallest vertebrate, is a tiny goby of one of the Philippine lakes of Luzon. Adult Pandaca pygmea are only a third of an inch in length and weigh about 0.00015 of an ounce. If you were to put 300 of them on a scale, they wouldn't equal the weight of an American penny. At less than half an inch, some male deep-sea anglerfishes are not much bigger, but what they lack in size they make up for in the sheer audacity of their mode of existence. On finding a female, males of some deep-sea anglerfish species latch their mouths onto her body and stay there for the remainder of their lives. It doesn't matter much where they fix their bite on the female, it could be on her abdomen or her head, they eventually become fused to her. Many times smaller, the male resembles little more than a modified fin, living off her blood, supply and fertilizing her intravenously. One female may end up with 3 or more males sprouting from her body like vestigial limbs. It looks like a lurid form of sexual harassment. Scientists have called it sexual parasitism. But the origins of this unconventional mating system are not so ignoble. It is estimated that female deep-sea anglerfishes occur at a density of about 1 per 800,000 cubic meters of water, which means a male is searching for a football-sized object in a darkened space about the volume of a football stadium. Thus, it is desperately hard for anglerfishes to find each other in the vast darkness of the abyss, making it wise to hang on to your partner if you find one. At the time that Peter Greenwood and J. R. Norman revised A History of Fishes in 1975, no free-swimming adult male anglerfish had been found, leading ichthyologists to speculate that the only alternative to successful latching is death. But the University of Washington's Ted Peach, curator of fishes at the Burke Museum of Natural History and Culture and the world's leading authority on deep-sea anglerfishes, tells me that there are now hundreds of free-living males in specimen collections around the world. In exchange for the male being the ultimate couch potato, the female never has to wonder where her mate is on a Saturday evening. It turns out that some males do indeed amount to little more than an appendage. Another fish superlative is their fecundity, which is also unmatched among vertebrates. A single ling, 5 feet long and weighing 54 pounds, had 28,361,000 eggs in her ovaries. Even that pales compared to the 300 million eggs carried by an ocean sunfish, the largest of all bony fishes. That such a grand creature can be the product of such a paltry parental investment as a teeny egg released into the water column might contribute to the common bias that fishes are unworthy of our consideration. But it bears reminding that all living things start from a single cell. And as we'll see in the section on parenting styles, parental care is well developed in many fishes. From its humble beginnings as an egg smaller than this letter O, a mature adult ling can grow to be close to 6 feet long, and it is another superlative of fishes that they can increase so much in size from the start of their independent life cycle. But the growth champion among vertebrates may be the pointed-tailed ocean sunfish. While not streamlined, they grow from 1/10 of an inch to 10 feet in length, and can weigh 60 million times more as an adult. Sharks lie at the opposite end of the spectrum of fish fecundity. Some species reproduce at a rate of only 1 baby a year. And that's only after they reach sexual maturity, which for some species can take a quarter century or more. In parts of their range, spiny dogfish sharks, a heavily fished species that you might have dissected in a college biology course, average 35 years old before they are ready to breed. Sharks have a placental structure as complex as that of mammals. Pregnancies are few and far between, and gestation can be lengthy. Frilled sharks carry their babies for over 3 years, the longest known pregnancy in nature. I sure hope they don't get morning sickness. Dogfishes can't fly, nor can any other fishes, but they just might be the world's superlative for gliding. Best known of these are the flying fishes, of which there are about 70 species inhabiting the surfaces of the open ocean. Flying fishes have greatly enlarged pectoral fins that function as wings. In preparation for launch, they can reach speeds of 40 miles per hour. Once airborne, the lower lobe of the tail may be dipped into the water and used as a supercharger to extend flights to 1,200 feet or more. Flights are usually just above the surface, but sometimes gusts of wind carry these aerialists 15 to 20 feet high, which may explain why they sometimes land on ship decks. I wonder if the respiratory limitations of being a water breather have kept flying fishes from becoming truly flapping their wings for fully sustained flight? Fishes of several other types also launch themselves into the air, including the characins of South America and Africa. And, never mind that their name sounds more like a circus act, the flying gurnards. Speaking of superlatives and names, surely one of the longest belongs to Hawaii's state fish, the rectangular triggerfish, known by the locals as Homohomonukunukwapue. Perhaps the award for least flattering name should go to an anglerfish dubbed the hairy-jawed sackmouth, and for most preposterous to the sarcastic fringehead. For the title of crudest, I nominate a small coastal dweller, the slippery dick. But really, the most exciting breaking news on fishes is the steady stream of discoveries on how they think, feel, and live their lives. Scarcely a week now passes without a revealing new discovery of fish biology and behavior. Careful observations on reefs are uncovering nuanced social dynamics of cleaner, client-fish mutualisms that defy the human conceit that fishes are dim-witted pea-brains and slaves to instinct. And the notorious 3-second fish memory has been debunked by simple laboratory investigations. In the pages ahead, we'll explore how fishes are not just sentient, but aware, communicative, social, tool-using, virtuous, even Machiavellian. Lowly not. Among the vertebrate animals, mammals, birds, reptiles, amphibians, and fishes, it is the fishes that are the most alien to our sensibilities. Lacking detectable facial expressions and appearing mute, fishes are more easily dismissed than our fellow air breathers. Their place in human culture falls almost universally into two entwined contexts: something to be caught and something to be eaten. Hooking and yanking them from the water has not just been seen as benign but as a symbol of all that's good about life. Fishing appears gratuitously in advertising, and the logo of one of America's most beloved film production studios, DreamWorks, features a Tom Sawyer-esque boy relaxing with a fishing pole. You may have met self-professed vegetarians who nonetheless eat fishes, as if there were no moral distinction between a cod and a cucumber. Why have we tended to relegate fishes beyond the outer orbit of our circle of moral concern? For one thing, they are cold-blooded, a layman's term with little credibility in science. I do not see why having a built-in thermostat or not should have anything to do with an organism's moral status. In any event, most fishes' blood does not run cold. Fishes are ectothermic, meaning that their body temperatures are governed by outside factors, notably the water they are living in. If they live in warm tropical waters, their blood runs warm. If they live in the frigid reaches of the ocean depths or the polar regions, as many fishes do, then their body temperatures hover around freezing. But even that description falls short. Tunas, swordfishes, and some sharks are partly endothermic. They can maintain body temperatures warmer than their surroundings. They achieve this by capturing heat generated by their powerful active swimming muscles. Bluefin tunas keep their muscle temperatures between 82 and 91 degrees Fahrenheit in waters ranging from 45 to 81 degrees. Similarly, many sharks have a large vein that warms the central nervous system by draining warm blood from the core swimming muscles to the spinal cord. The large, predatory billfishes use this heat to warm their brains and eyes for optimal functioning in deeper, cooler waters. In March 2015, scientists described the first truly endothermic fish, the opa, which maintains its body temperature at about 9 degrees Fahrenheit above the cold waters it swims in at depths of several hundred feet. Thanks to heat generated by the flapping of its long pectoral fins and conserved by a countercurrent heat exchange system in its gills. Another prejudice we hold against fishes is that they are primitive, which in this context has a host of unflattering Connotations: simple, undeveloped, dim, inflexible, and unfeeling. Fishes were born in front of my sunrise, wrote D.H. Lawrence in his 1921 poem Fish. No one is questioning that fishes have been around a long time, but therein lies the fallacy of labeling fishes as primitive. This bias presumes that those that stayed in the water stopped evolving the moment a few of them went ashore, a notion completely at odds with the tireless process of evolution. The brains and bodies of all extant vertebrates are a mosaic of primitive and advanced characteristics. Given time, and there's been plenty, natural selection keeps what works and winnows the rest, mainly through a process of gradual refinement. All of the fish species that were living at the dawn of legs and lungs are long gone. About half of the fishes we see on the planet today belong to a group called the Percomorpha, which underwent an orgy of speciation just 50 million years ago and reached peak diversity around 15 Maya, when the ape family, Hominoidea, to which we belong, was also evolving. So about half of fish species are no more primitive than we are. But the descendants of early fishes have been evolving eons longer than their terrestrial counterparts, and on these terms fishes are the most highly evolved of all vertebrates. You might be surprised to know that fishes have the genetic machinery to make fingers, something that shows how similar fishes are to modern mammals. They just don't develop fingers, but fins instead, since fins are better for swimming than fingers are. And don't forget your segmented musculature. The rectus abdominis, the washboard stomach that graces the torso of our fittest athletes, harkens back to the axial muscle segmentation first laid down by the fishes. As the title of Neil Shubin's popular book Your Inner Fish reminds us, our ancestors were early fishes, and our bodies are packed with modified structures traceable to those of our common aquatic forebears. An older organism isn't necessarily simpler. Evolution does not trend relentlessly toward increased sophistication and size. Not only were the largest dinosaurs much larger than modern reptiles, paleontologists have recently unearthed evidence that they were social creatures with parental care and modes of communication at least as complex as those of modern reptiles. Similarly, the largest terrestrial mammals died out thousands or millions of years ago, at a time when mammalian diversity flourished. The true age of mammals is over. We tend to think of the last 65 million years as the age of mammals, but teleost fishes have been diversifying much more during that time. The age of teleosts may not sound quite as sexy, but it's more accurate. Just as evolution does not proceed inescapably toward increased complexity, nor is it a process of perfection. For all the elegance with which adaptations allow animals to function optimally, it is a fallacy that animals are perfectly tailored to their environments. They can't be, because environments aren't static. Weather patterns, geological shifts such as earthquakes and volcanoes, and the constant process of erosion present moving targets. Even beyond these instabilities, nature is not fully efficient. There are inevitably compromises. Human examples include our appendix, our wisdom teeth, and the blind spot where the optic nerve interrupts the retina. For fishes, there is the closing of the gill covers necessary for respiration, which causes a forward thrust. If a fish wishes to remain stationary, as a resting fish usually does, she needs to compensate for the gill thrust. This is why you will rarely see a stationary fish whose pectoral fins are not in motion. As we learn more about fishes, be it their evolution or their behavior, our capacity to identify with them grows, along with our ability to relate their existence to our own. Central to empathy, the capacity to place oneself in another's shoes, or in this case, fins, is an understanding of the experiences of the other. Central to that is an appreciation of their sensory worlds. Part 2. What a fish perceives. There is no truth. There is only perception. Gustave Flaubert. What a fish sees. Red gold, water precious, mirror flat bright eye. From Fish, by D.H. Lawrence. We are taught that there are 5 senses: vision, smell, hearing, touch, and taste. In truth, this is a restricted list. Think how dull life would be if you did not have a sense of pleasure. And while the idea of life without pain is appealing, how dangerous would it be if you didn't realize you were resting your hand on a burning hot stove? Without a sense of balance, we'd have no success walking, let alone bicycling. Without the ability to detect pressure, handling a knife and fork adeptly would become tasks requiring Herculean feats of concentration. As we might expect for creatures who have had a lot of time to evolve, fishes have diverse, advanced modes of sensory perception. One of my favorite concepts learned as a student of animal behavior is Umwelt, a term created early in the 20th century by the German biologist Jacob von Uexküll. You can think of an animal's Umwelt as its sensory world. Because their sensory apparatus varies, different species may have different perceptions of the world even if they inhabit the same environment. For instance, owls, bats, and moths all fly in the nighttime, yet differences in their biology predict differences in the Umwelt for each. Owls rely mainly on vision and hearing to catch their prey. Bats also depend on hearing, but in a way that is quite different from the owl. They interpret echoes of their own high-pitched calls, hunting and navigating. By echolocation. Moths, as invertebrates, may be the least relatable of the three from the perspective of our own Umwelt, but we do know that they have good vision and that they can find mates over long distances with their superb perfume detectors. How a species' senses work goes some way toward understanding the mysteries of its felt experiences. We can expect fishes' Umwelts to differ from ours since they evolved in water and not air. But evolution is a conservative designer with a tendency to hang on to a neat idea. Case in point, fish eyes. Apart from their obvious lack of eyelids, fishes' eyes resemble our own. Like most vertebrate eyeballs, including humans, a fish's eyeballs are served by 3 pairs of muscles that swivel the eye on all axes, as well as a suspensory ligament and retractor muscles that help the fish focus on those bubbles dancing up from the aerator. Or that upright creature staring intently from the other side of the glass. As the evolutionary forebears of land-dwelling animals, early fishes originated this system of seeing. It is not easy to spot the swiveling eye movements of most small fishes, but peer closely next time you visit an aquarium and you should be able to detect eye movements in the larger individuals as they shift their gaze to look at different parts of their surroundings. With a spherical lens of high refractive index, defined as the ratio of the speed of light through a medium to its speed through a vacuum, a fish can see as clearly underwater as we can see in air. Needless to say, fishes have neither lacrimal glands, nor tear ducts, nor eyelids to moisten the eye's delicate surface. They don't need them, since the eyeball is kept constantly clean and moist by the water they swim in. Seahorses, blennies, gobies, and flounders have further upgraded their eye musculature to allow each eye to rotate independently, as in chameleon lizards. I can only conclude from this that a creature thus endowed is able to process 2 visual fields at a time. That seems just so radically different from what human brains do, and when I try to imagine the mental experience of 2 independent visual fields, each under my conscious control, it exceeds my Umwelt no less than trying to imagine a limit to the universe. Although a team of scientists from Israel and Italy have simulated the visual system of chameleons by building a robotic head with 2 independently moving cameras, I am not aware of any attempts to understand how a single brain processes them. I saw chameleon having twat hots simultaneously as one eye focuses on a juicy grasshopper on the neighboring twig while the other eye searches the branches overhead for a better approach route? Can a seahorse ogle a potential mate with one eye while tracking the movements of a lurking predator with the other? My single-track brain can't. If I read the newspaper while the radio plays This American Life, my mind can toggle back and forth between the two, but try as I might I cannot stream both stories at the same instant. I also have trouble getting my head around the visual experience of flounders, especially during their early childhood. Baby flounders look like any other normal fish, swimming upright with one eye on each side. Then, in preparation for adult life, they undergo a bizarre transformation. One eye migrates to the other side of the face. It's like facial reconstructive surgery, only in slow motion and without scalpels and sutures. It isn't even always slow. The entire migration takes just 5 days if you're a starry flounder, and less than 1 day in some species. If a fish can have an awkward adolescence, this one qualifies. In exchange for the indignity of having both eyes nestled next to each other on one flank, flounders have fabulous binocular vision. Like proud neighbors, the two eyes protrude from the body, and each can swivel independently. Binocular vision is a useful adaptation for a lifestyle of lying in wait on the sandy or stony bottom, exquisitely camouflaged against the substrate, watching for an opportunity to snatch an unsuspecting shrimp or other unfortunate passerby with a lightning-fast lunge. With refined depth perception, a flounder can better judge the timing and wisdom of her ambush. Ocular migration has obviously proven an effective survival strategy for flounders and related flatfishes, of which there are more than 650 species, including sols, turbots, halibuts, sand dabs, plaices, and tonguefishes. Some species are referred to as right-eye flounders, always lying on their left side after their left eye migrates to the right side of the body. Others are left-eye flounders. Despite their fine adaptations, many Atlantic flounder and sole species are now threatened by overfishing. The four-eyed fish, which inhabits fresh and brackish waters along the Atlantic coast of Central and South America, enhances its visual field in a different way. Nature's inventors of the bifocal lens, these relatives of the guppy sport a discrete demarcation between the upper and lower portion of their retina. The fish swims so that the demarcation aligns exactly with the plane of the water surface, the airborne portion of the eyes providing ideal air vision while the submerged portion accommodates the aquatic medium. Flexible genetic coding makes the upper eyes sensitive to green light wavelengths that predominate in air, and the lower eyes more sensitive to the yellow wavelengths found in muddy waters. This is a valuable visual toolkit when you want to search for a tasty morsel below without being surprise attacked by a hungry bird from above. Most larger, faster, open-ocean predatory fishes, including swordfishes, tunas, and some sharks, rely on speed and keen vision to catch prey. The eyes of a 12-foot swordfish can measure nearly 4 inches across. Yet hunting underwater presents a set of special visual challenges. If you've ever entered a cave without a flashlight, then you'll have a sense of what fishes experience as they dive deeper below the surface, where there is less light available to see with. There's another problem: water temperatures drop with greater depth, and cold retards brain and muscle function, delaying response times. To surmount the sluggish effects of cold, some fishes have evolved an ingenious means of improving the functioning of their brains and eyes. They harness heat generated by their muscles, which powers their sensory organs to perform at higher capacity. Swordfishes can heat up their eyes 20 to 30 degrees Fahrenheit above the water temperature. The heat is generated by a countercurrent exchange between the incoming and outgoing blood vessels surrounding the eye muscles. Arteries bringing cold blood from the heart and veins are warmed by a special heat-generating organ in one of the eye muscles. These arteries form a tight, lattice network, enhancing the exchange of heat between them. Studies of eyes removed from recently caught swordfishes suggest that this warming strategy improves by more than tenfold the fish's ability to track rapid changes in prey movements. Unlike swordfishes, many sharks prefer to hunt at nighttime, when light levels are exceedingly low. Supremely adapted to their realms, sharks' eyes have a layer of reflective cells called the tapetum lucidum next to the retina. Light hitting this layer bounces back through the shark's eye, striking the retina twice and effectively doubling the shark's night vision. This effect is what creates the familiar eye shine of cats and other terrestrial night stalkers. If sharks walked on land, you would see them in the headlights at night by the eerie glow of their eyes. Avoiding predators is no less a priority than is catching prey. Be it in an ocean, lake, or stream, fishes use a variety of visual techniques to get the upper fin. For those living in the shallows, for example, the underside of the water surface acts as a mirror. This enables a fish to see the reflection of objects that are not in direct view. A bluegill, a saucer-sized fish that lives in the shallows of North American lakes, ponds, and slow-moving streams, may be able to spy on a predatory pike lurking on the far side of a rock or thicket of pondweeds by gazing up at the surface reflection. What's good for the goose is also good for the gander, and I'd expect that predators may also use this technique to spy on their prey. I believe this could be studied quite easily in a temporary captive setting. The mirror technique that the bluegill uses only works in calm waters, and in such conditions fishes can also see quite well what is going on above the surface, allowing them to take evasive action when a diving bird approaches. The fact that wavy water impairs the ability to resolve objects above the surface might explain why seabirds hunt more often and catch more fishes in wavy than in calm waters. The refractive properties of calm water also enhance fishes' ability to see objects on the shoreline. Fishermen armed with this knowledge sometimes stand farther away from the water's edge to reduce the likelihood of detection by their quarry. Color badges and flashlights. There are times, of course, when being detected is the goal. Coral reefs present diverse opportunities for visual innovation. Corals grow in tropical seas at shallow depths, where temperatures and light levels are high. Light does magical things with color, which accounts for the mesmerizing kaleidoscope displayed on the bodies of reef fishes. In fact, when scientists in 2014 discovered evidence of rods and cones in a fossilized shark-like creature that lived 300 million years ago, they concluded that color vision was invented underwater. In the ages since, fishes have evolved visual capacities beyond our own. For example, most modern bony fishes are tetrachromatic, allowing them to see colors more vividly than we do. We are trichromatic creatures, which means we possess only 3 types of cone cells in our eyes, and our color spectrum is more limited. Having 4 types of cone cells, fishes' eyes provide 4 independent channels for conveying color information. Some fishes also see light in the near-ultraviolet spectrum, where light's electromagnetic wavelengths are shorter than what we can see in the so-called visible spectrum. This helps explain why about 100 known species from 22 families of reef fishes reflect large amounts of UV light from their skin. It all makes me wonder whether a fish gets more excited to see a diver whose wetsuit has blue and yellow racing stripes compared to one wearing a plain black wetsuit. In 2010, scientists made a discovery that illustrates the value of having a wider visual spectrum than someone else has. Their work focused on visual communication in damselfishes, a colorful and diverse group of reef denizens. They studied 2 species, the ambon damselfish and the lemon damselfish, which inhabit the same reefs in the western Pacific and which, to humans, look identical. Ambon damselfishes defend their territories most vigorously against members of their own species. But how do they know an intruder isn't merely a lemon damsel? The researchers had a hunch that vision was still somehow playing a role. It turns out each species has a different facial pattern visible only in the UV light spectrum. When researchers shone a UV light on them, the damsels' faces revealed attractive patterns of dots and arcs resembling a fingerprint, which differed between species in a subtle but consistent way. Tested for their recognition skills in captivity, the fishes could reliably indicate correct choices by tapping a picture of their own species with their mouths in return for a food reward. When the researchers used UV filters to eliminate this visual information, the fishes started failing the tests. Furthermore, because the predators of damselfishes appear blind to UV light, the damselfish's face recognition system operates covertly without compromising the camouflage that helps them avoid being seen by their finned foes. It's like being the only one to know who's behind that alluring mask at the masquerade ball. Fishes' bodies have a variety of ways of expressing themselves. Through color. In addition to species identification, the coloration of many fishes conveys information to their species mates about gender, age, reproductive status, and mood. Pigmented cells in the skin contain carotenoids and other compounds that reflect warm colors, yellow, orange, and red. White coloration is not produced passively, by a lack of pigment, but actively, by light reflected from crystals of uric acid in leucophores and guanine in iridophores. Greens, blues, and violets are mostly produced by structural patterns in a fish's skin and scales, and further varied by the thickness of these tissues. Think of a very colorful clownfish, whose coloration identifies him as a particular species of anemone fish and signals a conspicuous warning to other fishes that it would not be a good idea to follow him into the stinging tentacles of his home anemone. If wearing bright clothes is useful, being able to change them may be even better. By expanding or contracting their melanophores, clusters of cells containing black granules, fishes like cichlids and boxfishes are able to quickly turn darker or lighter in color. Some fishes, such as flounders and cornetfishes, have remarkable control over which cells expand or contract, while colorful coral reef fishes in particular can usually control the intensity of their so-called poster coloration. They can ramp up their beauty to lure a potential mate or intimidate the competition, or tone it down to mollify an aggressive competitor or go undetected by a predator. I think of the flatfishes as the champions of pigment manipulation. They use their skin to melt chameleon-like into the background. I remember flipping through a biology textbook when I was in high school and encountering a jaw-dropping photo of a flounder who had been placed on a checkerboard in his tank. Within minutes, the flatfish had produced a fine rendition of a checkerboard across his back. From a distance, the flounder effectively disappears. This ability to mimic backgrounds by changing the distribution of skin pigments is a complex and poorly understood process that involves vision and hormones. If one of the flounder's eyes is damaged or covered by sand, they have difficulty matching their colors to their surroundings, which hints at some level of conscious control by the flounder rather than a cellular-level mechanism. Surrounded befriend sandy nemes, fishes face a compromise between being detected and not being detected. Near the surface in the sunlight zone, practically everything is visible. But light penetration in water decreases exponentially with depth. Being seen is a high priority for a fish, for 90% living in the twilight zone between 100 and 1,000 meters have light-emitting organs that serve as beacons in the darkness. The proportion is even higher for fishes living in the midnight zone, the vast abyss at depths of 2,000 meters and beyond, where no light reaches. Fishes who live here include bristlemouths, lanternfishes, and the famous anglerfishes. Down here, most of the light is produced by luminous bacteria that coexist with the fishes in an ancient symbiosis. In return for room and board, the light-producing bacteria provide a range of benefits to their hosts. Deep-sea anglerfishes are experts when it comes to light displays. They emit light from the fishing lure that protrudes from their head, and in some species also from a tree-like structure suspended from the lower jaw. These glowing adornments enhance their attractiveness to potential prey who, drawn like moths to a candle, swim to their deaths in the jaws of these ambush predators. On the flip side, sudden bursts of light cast from the same structures can be used to startle would-be predators. Body lights can also provide camouflage by casting a faint glow across the fish's lower side, making her less visible against the dim light filtering down from above. And when fishes want to spend some time with companions, the distinctive light patterns produced by these organs can help in recognizing others of their own kind. Ponyfishes have a peculiar method of luminescence. The photophore, or package of light-producing bacteria, carried by males around their throat is shown inward toward a specialized swim bladder with a reflective coating. The light bounces off this coating and out through a transparent patch of skin. By controlling a muscular shutter in the body wall, the ponyfish creates a flashing display. Schools of males sometimes coordinate their flashing to create a dazzling show, which scientists believe is a strategy to get females into a mating mood. Flashlight fishes, one of the few bioluminescent fishes generally not found in deep waters, take a more direct approach to illumination, using a multifunctional light consisting of a semicircular organ just below each eye. This pair of organs contains luminescent bacteria whose continuously emitted light can be turned on and off by the fish using a muscular lid. Like ponyfishes, flashlight fishes gather in nighttime shoals where their combined light helps attract, as well as illuminate, zooplankton prey. These fishes also use the light to evade predators. When the danger approaches, the targeted fish keeps his light on until the last moment before switching off and changing direction. Mated pairs of flashlight fishes maintain territories over a reef, and if an intruding flashlight fish approaches, the female of the pair will swim up and flash her light literally in the interloper's face, as if to say, "Get lost." These deep-sea light shows happen in the blue-green spectrum, which is the color of most bioluminescence, probably because aquamarine light travels farthest through water. But there is one group of fishes that breaks the color rule, the loosejaws. Named for a capacious lower mandible whose flexible hinge allows an enormous gape, these fishes might as well have been called the stoplight fish, for the powerful beam of light they shine from a concentrated photophore beneath each eye is red. The color is achieved by a unique fluorescent protein in some species and by a simple gel-like filter over the photophores in others. Naturally, evolution has seen to it that loose jaws can see red, thanks to a small change in a gene responsible for eye pigment structure. The advantage is huge, a flashlight beam that only the bearer can see. Thus endowed, these hunters of the abyss can spy on others without being seen. Whereas other deep-sea fishes use their lights intermittently, flickering and flashing lest they be discovered and eaten, loose jaws audaciously keep their lamps lit full-time, invisible to their predators and to the prey they stalk with impunity. It is the deep-sea answer to night vision goggles. Fooled you! Clearly, fishes have a diverse and innovative visual repertoire. Their tools are used to enhance their seeing ability, to make themselves more or less visible, to declare their identities, to lure and repel, and to manipulate. But how do fishes perceive what they themselves see? What is the mental experience of a fish, and how might it compare to our own? One way of probing this question is by considering optical illusions. If an animal is unaffected by a visual image that fools us, then it would seem that that animal perceives visual fields in a mechanical way, as a robot might perceive them. If, however, they fall for the illusion as we do, it suggests that they have a similar mental experience of what they are seeing. In Alex and Me, Irene Pepperberg's touching memoir of 30 years with an African grey parrot, one of the many captivating findings reported is that these intelligent birds perceive optical illusions as we do, they are fooled by them. The implication, as Pepperberg remarks, is that parrots literally see the world as we do. Figure 1, the Ebbinghaus illusion. Are fishes fooled by optical illusions? Well, in a captive study of red-tail splitfins, small fishes that originate from highland Mexican streams, they learned to tap the larger of two discs to get a food reward. Once they had mastered the task, the scientists presented them with the Ebbinghaus illusion, which consists of two discs of the same size, one of which is surrounded by larger discs, making it appear smaller than the other, which is surrounded by smaller discs. The splitfins preferred the latter disc. This result showed the scientists that red-tailed splitfins do not perceive things in a mindless, stimulus-response way. Rather, they form mental concepts, sometimes fallible ones, based on their perceptions. Similarly, an earlier study found that red-tailed splitfins also fall for the more familiar Muller-Lyer illusion, in which two identical horizontal lines appear to have different lengths. Trained to choose a longer line, they chose the line labeled B. Figure 2, the Muller-Lyer illusion. Studies of goldfishes and bamboo sharks show that they, too, respond to visual illusions. You can train goldfishes to discriminate black triangles from black squares on a white background. Then if you present them with a Kanitsa triangle or a Kanitsa square, they perceive a triangle and a square, respectively. Kanitsa illusions were developed in the 1950s by the Italian psychologist Gaetano Kanitsa. When humans regard these figures, we see a white triangle that looks slightly brighter than the background, even though no triangle is actually drawn. So what the goldfish brains are doing is the same as what ours do, completing an incomplete picture. Figure 3, a Kanitsa triangle. That splitfins, goldfishes, and bamboo sharks can complete an incomplete picture isn't meant to imply that they are unique among fishes in falling for optical illusions. They were simply the species chosen for these studies. Splitfins and goldfishes are only distantly related, so it seems likely that many other fishes would be fooled by optical illusions. These species are studied for the mundane reason that their care in captivity has been well worked out, so using them is a matter of convenience. It takes time and effort to conduct meticulous studies of animals. So, what we know about fishes is only a tiny slice of what they know. In the survival game, fishes may exploit the visual perceptions of other fishes with illusions of their own. One way to do this is to deflect a predator's attack away from the important parts of one's body. For the fairly obvious reason that it is more likely to be lethal, predators usually direct an attack at the head end of their prey. That many aquatic predators tend to aim for the eyes is evidenced by the evolution of deceptive eye spots on many fishes. Examples of fishes who benefit from this deception include cichlids, butterfly fishes, angelfishes, pufferfishes, and bowfins. The deception may be enhanced in various ways. Like us, fishes are more likely to notice bright colors, so those deceptive eye spots tend toward conspicuous brilliance, while the real eye at the other end may be relatively obscure. The pattern of a young emperor angelfish doesn't include an eye spot, but a bullseye surrounded by concentric rings of alternating white and neon blue looks just as effective, while the real eye is obliterated by a maze of meandering lines. A predator rushing in for the kill won't have time to make fine assessments, and these color tricks might tip the scales in the prey's favor. A further enhancement is having a tail end shaped to resemble a fish's head. The rear end of a cometfish is so arranged to resemble the face of a parrotfish, and the actual eye is virtually lost amid a constellation of white spots covering the entire body, including the eye itself. A behavioral manipulation can enhance these effects even further. Scientists have observed 2 species of butterfly fish who switch gears and swim slowly backward at the first sign of trouble, then suddenly lurch into forward overdrive if a predator darts in. If they move fast enough, the predator may be snapping at empty water. Otherwise, a butterfly fish is more likely to live on if the chunk of missing flesh came from the tail than if it came from the head. I find it endearing that fishes perceive optical illusions as we do, and that they are tricked by the visual deceptions of their intended prey. It says something special about the perceptual world, the Umwelt, of another being that her mind should construct something that isn't actually there. It suggests the capacity for belief. Beliefs and perceptions can be exploited, and as we've already seen, fishes use a range of deceptions, visual and otherwise, to improve their chances at success. As highly visual creatures, we may be able to relate to the importance of having the keen vision that most fishes have. From childhood games, we know the disorientation of being blindfolded, and we look on with admiration at how well blind humans learn to cope with the challenges. It is doubtful that a blind fish would live long, even if he inhabits the midnight zone, where built-in lights rule supreme. But fishes are not solely dependent on seeing to make their livelihoods. Like us, they have evolved other senses to help them navigate life's demands. What a fish hears, smells, and tastes. The universe is full of magical things, patiently waiting for our wits to grow sharper. Eden Philpotts. Just as water influences the dynamics of vision, so it does for hearing, smell, and taste. Water is a superb conductor of sound waves, where they are almost 5 times longer than in air, as sounds travel 5 times faster in water. Fishes have benefited from this since the dawn of bones and fins, using sound for both orientation and communication. Water is also an excellent medium for diffusing water-soluble chemical compounds, and is well suited for the perception of smells and tastes. Fishes have separate organs for smelling and tasting, although the distinction is blurred because all substances are encountered in a water solution. As they did color vision, fishes probably invented hearing. Despite the common assumption that fishes are silent, they actually have more ways of producing sounds than any other group of vertebrate animals. None of these methods involve the main method of all the other vertebrates, the vibration of air against membranes. Fishes can rapidly contract a pair of vocal muscles to vibrate their swim bladder, which also serves as a sound amplifier. They have the options of grating their teeth in their jaws, grinding additional sets of teeth lining their throat, rubbing bones together, stridulating their gill covers, and even, as we'll see, expelling bubbles from their anuses. Some land-dwelling vertebrates get creative in producing non-vocal sounds, such as the drumming of woodpeckers and the chest-pounding of gorillas, but fishes' terrestrial cousins possess just 2 types of vocal apparatus, the syrinx of birds and the larynx of all the rest. With their versatile acoustic portfolio, fishes produce a veritable symphony of sounds, especially in the percussion section. Among the descriptors we have assigned to them are hums, whistles, thumps, stridulations, creaks, grunts, pops, croaks, pulses, drums, knocks, purrs, brrrs, clicks, moans, chirps, buzzes, growls, and snaps. So notable are the sounds of some fishes that we have named the fishes accordingly: grunts, drums, trumpeters, croakers, sea robins, and grunters. Having ears evolved for processing vibrations in air and not water, we were until recently deaf to most of the sounds fishes were making. It was only in the past century, as underwater sound detecting technology improved, that the list of acoustic fishes began to grow. And yet, as recently as the 1930s, scientists believed that fishes were deaf. This prejudice probably arose from the fact that fishes lack an external hearing organ. With our human-centric view of the world, such a lack could only mean one thing: no hearing. Now we know better. Fishes don't need ears, thanks to water's incompressibility, which is why water is an excellent conductor of sounds. It is not until we peer inside a fish that we find structures modified and recruited for producing and processing sounds. Carl von Frisch, the Austrian biologist famous for his discovery of the dance language of honeybees, was also a devoted student of fish behavior and perceptions. Decades before he became the co-recipient of the Nobel Prize in 1973 for his contributions to the emergence of ethology, von Frisch was the first to demonstrate hearing in fishes. In the mid-1930s, he devised a simple but ingenious study in his lab with a blind catfish named Zavrel. He did this by lowering a piece of meat on the end of a stick into the water near the clay shelter in which Zavrel spent most of his days. Having an excellent sense of smell, Zavrel would soon emerge from his hiding place to retrieve food. After a few days of this routine, von Frisch began to whistle just before delivering the food. 6 days later, he was able to lure Zavrel from his lair just by whistling, thereby proving the fish could hear him. This experiment, and others that followed, were critical to advancing our appreciation for the fish's umwelt. Zavrel belongs to an evolutionarily successful group called the Otophysi, which number about 8,000 species. They have evolved a specialized hearing apparatus called the Weberian ossicles, named for its discoverer, the 19th-century German physician Ernst Heinrich Weber. The ossicles are a series of small bones derived from the first four of a fish's vertebrae behind the skull. These bones have become separated from their parent bones, forming a chain linking the gas-filled swim bladder with fluid-filled spaces surrounding the inner ear. The apparatus aids hearing by acting as a conductor and amplifier of sound waves, in a manner similar to the middle ear ossicles of mammals. There are ways in which fish hearing surpasses our own. Most fishes hear in the range of 50 Hz to 3000 Hz, which is within our own range of 20 Hz to 20,000 Hz, but careful studies in captive and wild settings have now documented sensitivity to ultrasounds in the upper range of bad hearing, up to 180,000 Hz in American shad and Gulf menhaden. That is well above the upper human limit. It is believed to be an adaptation for eavesdropping on the ultrasonic sounds produced by dolphins, who prey on these fishes. At the other end of the hearing spectrum, fishes such as cods, perches, and plaices are responsive to infrasounds as low as 1 Hz. Nobody knows for sure why these fishes have evolved the ability to tune into super low sound, but the vast aquatic environments they live in provide clues. Water does not move randomly in oceans and large lakes. Global climate patterns generate currents, local weather patterns produce waves, and our moon's gravitational pull drives the constant heave-ho of ocean tides. Moving water also bumps up against cliffs, beaches, islands, reefs, coastal shelves, and other submerged barriers. All of these forces combine to create ambient infrasound. Biologists from the University of Oslo, Norway, think that fishes use this acoustic information for orientation during migrations. Think of it as a fish's equivalent of birds' use of celestial cues. Pelagic fishes may also detect changes in the surface wave patterns caused by distant land formations and different water depths. Sensitivity to infrasound has also been reported in some cephalopods and crustaceans, further evidence of its utility. Fishes' hearing sensitivity makes them vulnerable to human-generated underwater noise. For instance, the delicate hair cells lining the inner hearing apparatus become severely damaged by high-intensity, low-frequency sounds produced by air guns used in marine petroleum exploration. Intense noise produced by seismic airgun prospecting off the coast of Norway decreased the abundance and catch rates of cods and haddocks in the adjacent area. Some fishes can also detect rapid pulses of sound, discerning as individual beats what we hear only as a constant whistle, and they are proficient at sound directionality, distinguishing sounds from directly ahead versus directly behind, and from directly above versus directly below, perceptual tasks that our brains are less able to manage. That said, 99% of airborne sound energy is reflected off the water surface, so fishes, even if they're congregating close to the shore, are not likely to hear, say, a group of humans talking on a beach. However, airborne sounds transmitted through a solid object, such as an oar bumping against the side of a boat, are easily detected by fishes. This is why anglers sitting in a boat learn to be quiet, and why experienced shore fishermen wander a few yards inland before moving to a new spot. They know that the fish thereafter may detect vibrations transmitted through the ground. With ingenuity, we can hear them, too. Fishermen along the Atlantic coast of Ghana use a special paddle as a sort of tuning fork. By placing his ear against a paddle immersed in the water, an experienced practitioner can hear the grunts and whines of nearby fishes, and by rotating the flat plane of the paddle he can locate their whereabouts. A fish's keen hearing may also work in the angler's favor, for many fishes may not realize that the worm they hear up ahead is, unfortunately for them, wriggling on a hook. Whereas migration and predator avoidance are useful functions for fishes listening, most sound production has a social function. Here's an example from piranhas. When the biologists Eric Parmentier from the University of Liege, Belgium, and Sandy Millo from the University of Algarve, Portugal, placed hydrophones in a tank holding captive red-bellied piranhas, they recorded a variety of sounds, 3 of which are common enough to have been ascribed possible functions. 1. A repetitive grunt or bark appears to signal a challenge to others. Another, a low thud, is usually made by the largest fish in the group during aggressive behavior and fighting. These 2 sounds are produced by a fast-twitching muscle next to the swim bladder that contracts 100 to 200 times per second. A third sound occurs when a piranha grinds or rapidly snaps her teeth while chasing another fish. These descriptors hint at a mean-spirited animal, befitting the piranha's pugnacious reputation as a savage devourer of living victims. In fact, piranhas are mostly scavengers and pose little danger to humans. Given that fishes use sounds to communicate with one another, might they also use sound to communicate with us? I know of no scientific study to test this, but there are many anecdotes. Karen Cheng, a computer scientist from the Washington, D.C. area, has 4 rescued goldfishes in a 20-gallon tank who, she claims, communicate with her at mealtimes. Around feeding time, when Karen or her husband is in the room but not paying attention to them, their goldfishes rise to the surface and make loud smacking sounds with their mouths. They will also hurl their bodies and bang their tails against the aquarium wall, apparently to get their humans' attention. The sounds produced can be heard from the other side of the room. They stop doing it when someone approaches the tank. They seem aware of us, says Karen. Whenever we come up to the tank, they abort their activities and swim over to the glass. They don't ignore you like the aquarium fish at the doctor's waiting room. Sarah Kindrick, a clinical protocol administrator with the National Institutes of Health, saw similar behavior in an 8-inch pink-tailed triggerfish who lived with her for about 3 years. Furchbar, as she named him, would take a pebble in his mouth and wrap it on the glass wall of his tank around the time she would normally feed him. This is not just an example of interspecies communication by a fish, it is tool use. Concerto in D major for fish. A further testament to fish's acute hearing is their ability to discriminate tonal patterns of sound, specifically music. Ava Chase, a research scientist at Harvard University, was interested to see if fishes could learn to categorize sounds as complex as music. She conducted an experiment using 3 pet store-bought koi named Beauty, Aro, and Peppy. Chase set up a sophisticated apparatus in the fish's tank that included a speaker at the side for presenting sounds, a response button on the bottom that fishes could push with their bodies, a light that signaled to the fish that his response had been recorded, and a nipple near the surface that dispensed a food pellet when the fish swam up and sucked it after a correct response. She then trained the fishes by rewarding them when they responded to a certain genre of music and not rewarding them for responding while another genre emanated from the speaker. She found that the koi were not only able to discriminate blues recordings from classical recordings, but that they could generalize these distinctions when presented with new artists and composers for each genre. For example, once familiar with the blues of Muddy Waters, the koi recognized its commonality with blues artist Coco Taylor, as they did for the classical music of Beethoven with that of Schubert. One of the three fishes, Aro, had an especially good ear, able to discriminate melodies in which timbre cues had been removed, that is, all the notes had the same quality except their pitch and timing. Chase concludes, it appears that can discriminate polyphonic music, playing multiple notes simultaneously, discriminate melodic patterns, and even classify music by artistic genre. Despite their skill as music connoisseurs, neither koi nor goldfishes were known by scientists to communicate using sounds. So it remains a mystery why a mute fish would have such discerning acoustic skills, though as we saw earlier there are benefits to being able to tune into ambient sounds in one's environment. Being able to discriminate subtle qualities in music is one thing, but it makes me wonder, what psychological effect might it have on a fish? Do fishes appreciate music, or is it just a neutral stimulus? A research team from the Agricultural University of Athens decided to investigate. They divided 240 common carps among 12 rectangular tanks and randomly assigned them to 3 different treatments: no music, Mozart's Romance, Andante for Mein Klein-Nacht music, and the anonymous 19th century romanza, Juanterdis, a name it got from its use in the 1952 French film Forbidden Games. The track duration for these pieces was 6:43 and 2:50, respectively, and the assigned fishes were exposed to 4 hours of it a day for 106 days. Music exposure was done on weekdays only. Like office workers, the fishes got the weekends off. Fishes in both music groups grew faster than the control group. Feeding efficiency, growth rates, and weight gains were higher with either of the 2 romantic music recordings than without, and intestinal function appeared to be improved. When these fishes were presented with noise or with non-musical human sounds, the research team found no such changes. It is a central challenge of animal studies that the subjects cannot report to you in plain language how they are feeling. With these data, we can only speculate that carps are responding positively or negatively to the music. For instance, a skeptic might suggest that the fishes grew stronger from trying to escape the incessant sound of violins and oboes. I must say that, much as I enjoy classical music, hearing the same track over and over is not my idea of acoustic heaven. We should also consider the possibility that the fish's growth was no reflection of any subjective experience, but a mechanical response to a physical stimulus. An earlier study by the same Greek scientists noted favorable responses in response to Mozart by gilt-head sea bream, a species with very limited, lo-fi hearing. We also should be wary of anthropomorphism, for there may be little basis for assuming that what we perceive as pleasant music is perceived that way by a fish. Perhaps they prefer any sound to none at all. On that score, a better control than silence would be a recording of non-musical sounds. There are studies dating back a century that find human patients reporting improved relaxation and reduced pain when exposed to music that they enjoy. A 2015 review of 70 clinical trials involving more than 7,000 patients concluded that music was an effective therapy before, after, and even during surgery, and that it reduces patient anxiety and the need for painkillers. My point here is that music, or more generally, patterned, tonal sounds, may tap deeply into our biology with therapeutic results. The implication is that music appreciation might be widespread in nature. When I asked one of the authors of the Greek studies, the biologist Nafsika Karakatsouli, she expressed uncertainty that carps enjoy music. I am not at all convinced that music may have substantial positive effects for fish. There is no music underwater. However, there are plenty of other natural sounds, more relevant to fish living underwater, that may have some meaning to fish and may have produced better results. Nonetheless, some of the fish species we examined, especially carp, did perform better when music was transmitted. Karakatsili agrees that a better approach would be to see if carps would choose by themselves an environment with music or not. There isn't anything musical about the sounds that herrings make, but their innovative method might warrant a Fish Grammy Award. One paper describes the first example of what might loosely be termed flatulent communication. Both Pacific and Atlantic herrings break wind by releasing gas bubbles from the anal duct region, producing distinctive bursts of pulses, or what the research team playfully named fast repetitive ticks. A bout of FRTs can last up to 7 seconds. Try that at home. The gas probably originates in the gut or the swim bladder. It isn't clear how these sounds function in herring society, but since per capita rates of sound production are higher in denser schools of herrings, a social function is suspected. So far, there is no evidence that herrings ever beg your pardon. I couldn't think of a better segue from fish's sense of hearing to their sense of smell than herring FRTs. So let us have a sniff at their smell and taste. A good smell. You may think that a dead fish smells bad, but living fishes have a good sense of smell. They use chemical cues for finding food, finding mates, identifying danger, and homing. Smells are especially useful in aquatic environments where murky conditions make vision unreliable. Some fishes can recognize others of their own kind by scent alone. Sticklebacks, for example, use smell to identify mates of their own species, where proximity to another stickleback species might otherwise present the risk of mating with the wrong kind. The sophistication of the smelling organs of fishes varies greatly, but the basic design is shared among all the bony fishes. Unlike other vertebrates, fishes' nostrils do not do double duty as organs of smell and openings for breathing, they are used exclusively for smell. Each nostril is populated by layers of specialized cells composing the olfactory epithelium, which is folded upon itself to save space, forming a rosette. Some fishes expand and contract their nostrils, and thousands of tiny cilia pulse in sequence to propel water into and out of the sense organ. Signals from the epithelium are sent to the olfactory bulb at the front of the brain. Smell is an extremely useful sense for some fishes, as evidenced by their legendary sensitivity. A sockeye salmon can sense shrimp extract at concentrations of 1 part to 100 million parts water, which translates in human terms to 5 teaspoons in an Olympic-sized swimming pool. Other salmon can detect the smell of a seal or sea lion diluted to 1/80 billionth of water volume, which is about 2/3 of a drop in the same pool. A shark's sense of smell is about 10,000 times better than ours. But the champion sniffer among all fishes is the American eel, which can detect the equivalent of less than 1/10 millionth of a drop of their home water in the Olympic pool. Like salmons, eels make long migrations back to specific spawning sites, and they follow a subtle gradient of scent to get there. One of fish's most useful adaptations is the production of an alarm chemical in the presence of danger, such as a predatory fish or a spear fisherman. Once again, we owe it to Carl von Frisch for discovering yet another phenomenon in the world of fish senses. When he accidentally injured one of his captive minnows, von Frisch noticed that other fishes in the tank began darting back and forth and freezing in place, classic predator evading behavior. Experiments by von Frisch and others showed that injured minnows release a pheromone, a secreted or excreted chemical factor that triggers a social response in members of the same species. Detecting this particular pheromone causes agitated reactions by the minnows. Von Frisch coined the term Schreckstoff for these pheromones. The cells that release Schreckstoff are located in the skin and are fragile enough that they will rupture and release the substance if a fish is placed on moist paper. And it is potent stuff, 1/1000th of a milligram of chopped skin is enough to elicit a fright reaction from another fish in a 3.7-gallon aquarium. That's like chopping a marshmallow into 20 million pieces, dropping one piece into a sink full of water, then trying to taste the sweetness. Schreckstoff must have evolved long ago, for it is produced by several families of bony fishes. As a freely available signal, Schreckstoff acts like a fire alarm that can be used by other nearby fishes, including different species that may also recognize it. Case in point, fathead minnows. When they smell the poop of northern pikes who have fed on other fathead minnows or on brook sticklebacks, both of which produce Schreckstoff in their skin, they immediately flee to hiding places or form tighter shoals. But if the pikes have been fed only on swordtail fishes, which do not produce Schreckstoff, the minnows show no signs of fear. Thus, it is not the smell of the pike that the minnows are reacting to. Instead, they detect and react to the Schreckstoff from the pike's victims. It is probably due to olfactory skills like the minnows that pikes refrain from defecating in their own hunting grounds. The Schreckstoff reaction illustrates how fishes can extract subtle clues from waterborne chemicals, but Schreckstoff is not the only way to detect a fish foe by fragrance. There is the old-fashioned way of simply recognizing the smell of the predator. Juvenile lemon sharks react to the odor of American crocodiles, who sometimes prey on them. If you are an Atlantic salmon, it depends on what your predator has been eating. In a study conducted at Swansea University in Wales, predator-naive juvenile salmons were presented with water containing traces of dung from one of their natural enemies, the Eurasian otter. The salmons only showed a fear response if the otter had been dining on salmon. In those cases, they fled the smell, then remained still, and they breathed faster. Salmons exposed to blank water or to dung from otters on a non-salmon diet were unfazed. The scientists concluded that Atlantic salmons apparently do not innately recognize otters as a threat, they perceive them as a danger only if salmon is on the menu. This generalized mode of predator detection works well because it does not require learning the smell of different predators. Instead, one may just learn to recognize who has been eating one's own kind. If avoiding predators has a rival in the survival game, one candidate is the quest for sex. Just as aromas have been found to play an important role in human sexual attraction, so, too, do sex pheromones get a fish's juices flowing. For one, they help fishes identify who else is in a mating mood. Fishes have the ability to tease out subtle sex cues and use them for personal gain. Experiments from the 1950s showed that male frillfin gobies will start their courtship displays when a sample of water from a tank holding a sexually receptive female frillfin goby is added to their own tank. Later studies show that females are no less perceptive or active in the mating game. Female sheepshead swordtails from Mexico can discriminate the smell of well-fed males from hungry males of their species, 2 to 3 inch denizens of tropical rapids, and you can probably guess which they prefer, all else being equal. A well-nourished fish is a more resourceful one, which makes him the better sperm donor. Female swordtails do not discriminate the odor of well-fed females from hungry females, suggesting they are responding to male sex pheromones and not merely to food-based excretions. So far, we've been examining fish sensory systems as separate units, but they need not work in isolation. Male deep-sea anglerfishes illustrate the interplay of senses. They have the largest nostrils relative to head size of any animal on Earth, according to Ted Peach, the world's go-to guy on anglerfishes. His book Oceanic Anglerfishes is an astonishingly detailed and lavishly illustrated source for everything currently known about these bizarre fishes. The male anglerfish's nostrils are not his only well-developed senses, his eyes are also well put together, and Peach believes the two senses, smell and vision, function in tandem to help males find females in the dark abyss. A female releases a species-specific pheromone, and a male's fine sense of smell helps guide him toward her species-appropriate perfume. This is important because there are at least 162 known species of anglerfishes cruising about in the world's largest habitat, and you don't want to mate with the wrong one. When he gets close enough to the female, the male angler can confirm that she is his type by the signature of light she emits with the aid of glowing bacteria living in her filamentous lure. One can almost imagine a time in the deep past when the god of deep-sea anglerfishes proclaimed, "Let there be light," and a lot of the guesswork was taken out of finding a mate. One last note on the olfactory behavior of fishes. It has been widely assumed by the conservative-leaning scientific establishment that fishes' release of chemicals for communication is passive and not consciously controlled, since they lack external scent glands or scent-marking behavior. That's a shaky presumption. Consider a 2011 study of our friends, the sheepshead swordtails. In their fast-flowing habitats, males use at least two tactics to make females aware of their pheromones. They urinate more often when they have an audience of females, and when courting they situate themselves just upstream of females. For better or worse, that implies that in addition to being able to smell a male's sexual readiness, female sheepshead swordtails can also taste it. What else might a fish be tasting? Tasteful fishes. For fishes, the sense of taste is used mainly for food recognition. As for all the other main vertebrate groups, amphibians, reptiles, birds, and mammals, the primary organs of taste are taste buds. Fishes also display a range of teeth types, 8 in all, including incisors for snipping, canines for stabbing, molars for grinding, flattened triangular teeth for slicing, and teeth fused into beaks for scraping algae off corals. Like us, fishes have tongues and gustatory receptors connected to specialized nerves that relay taste signals to the brain. Not surprisingly, most of a fish's taste buds are located in the mouth and throat. But because fishes are quite literally immersed in the medium they smell and taste, many also have taste buds on other parts of their bodies, most often the lips and snout. Taste buds are also more numerous in fishes than in any other animal. For instance, a 15-inch channel catfish had approximately 680,000 taste buds on his entire body, including fins, nearly 100 times the human quota. These and other fishes of murky waters taste their way around their environments. Cave fishes also benefit from a bonanza of taste buds, which provide a high-definition flavor sensing system to aid food finding in the darkness. Many bottom feeders, including catfishes, sturgeons, and carps, are equipped with barbels, whisker-like feelers usually located around the mouth, which serve as taste sensors. In case you're wondering why fishes would need a sense of taste, well, they need it for the same reasons we do. Fishes have food preferences that may be distinctive to species and even individuals. It can take a fish a while to ascertain the palatability of a food item. If you've watched fishes in aquariums, you might have seen how they will sometimes take a morsel into their mouth, spit it out, then re-ingest it several times before either swallowing or rejecting it. Overall taste preferences within a fish species, and within different populations of the same species, generally do not vary much, as is the case for human ethnic groups. The same does not hold true of individual preferences. In our case, think Brussels sprouts, spicy versus mild, and the dizzying array of present-day variations on a cup of coffee. Studies of rainbow trouts and carps find that finicky eaters are not rare. Fish's reactions to unpleasant tastes are reminiscent of our own. Just as we will quickly eject a mouthful when we bite into a surprisingly rotten piece of fruit or meat, a Dover sole expresses her distaste for a food item by violently turning and rapidly swimming away from it, shaking or nodding her head. Stephen Reeves, the author of Fish Behavior in the Aquarium and in the Wild, describes a fish's reaction to the taste of toad tadpoles, a toxic and particularly foul-tasting item in its environment. It must be said that a very hungry bass, its back to the wall, will stoop to eat toad tadpoles. But if the reaction of other fishes that mistakenly take tadpoles in their mouth is anything to go by, they violently shake their head and you can almost see the grimace on their face. Having tadpoles on the menu is no great culinary experience for a fish. Living in a relatively dense aqueous medium imposes some limitations, but it also provides fishes with sensory opportunities unavailable to land-dwelling beasts. Can you imagine chatting with your neighbor using electric pulses? In the next part of the book, we'll get beyond the mainstream senses into some less familiar ways fishes use to perceive their environments. Navigation, touch, and beyond. When one flesh is waiting, there is electricity in the merest contact. Wallace Stegner. Fishes need to move about to meet their needs, and they must be in certain places at certain times if they are to be successful at making a living and making more fishes. Like us, fishes return to specific places at different times of day, such as feeding locations, hiding and sleeping places, and cleaning stations. At certain times of the year they return to mating, spawning, and nesting sites. Living in a complex volumetric habitat, fishes face a challenging spatial milieu. Fishes are excellent navigators, and they use a variety of methods to find their way around over both short and long distances. Blind cavefishes live in relatively small cave habitats, but most also live in total darkness, so having good navigation skills is important to them. These little fishes can learn the order of a sequence of landmarks en route to a destination by feeling the turbulence reflected off underwater obstacles. Swordfishes, parrotfishes, and sockeye salmons use sun compassing, setting their direction based on the angle of the sun. Yet others can use dead reckoning, taking wandering, novel outbound exploratory trips from a point of reference and then returning to home base using a direct path. The navigation feats of salmons are the stuff of legend. Being able to return to their natal streams to spawn after spending years in the open ocean ranks these anadromous fishes as having one of nature's finest built-in global positioning systems. As far as we know, this system uses at least 2 and possibly 3 sensory tools to work at full capacity: geomagnetic sense, smell, and possibly vision. Like sharks, eels, and tunas, these long-distance fishes plug into the Earth's magnetic field to aid their navigation. This manifests at the cellular level. Single cells containing microscopic magnetite crystals act like compass needles. By isolating cells from the nasal passages of trouts and exposing these cells to a rotating magnetic field, a research team from Germany, France, and Malaysia found that the cells themselves rotated. The magnetite particles are firmly attached to the cell membrane, and by constantly pulling toward magnetic field lines, these particles generate torque on the cell membrane when the salmon changes direction. That torque must be directly transmitted to stress-sensitive transducers of some kind, because evidence shows that the salmons can feel it. They also use their prodigious sense of smell. When heading downstream to the ocean, young salmons record the water chemistry along the way. Years later, they retrace their paths, following the distinctive odor signature of their home stream, like walking a trail in reverse. Anosmic salmons, whose noses had been experimentally plugged by biologists to eliminate their ability to smell, showed up in random streams, whereas unmolested fishes returned to their home streams to spawn. In a less invasive experiment, the same research team, led by the late Arthur Hassler of the University of Wisconsin, split a group of young coho salmons into 2 groups, each of which was exposed to one of 2 different, innocuous but fragrant chemicals, morpholine and phenylethyl alcohol. After this exposure period, salmons from both groups were released together directly into Lake Michigan. During the salmon spawning migration 1.5 years later, the researchers dripped morpholine into one stream and PA into another located 5 miles away. Nearly all of the recaptured salmons in the morpholine-scented stream were from the morpholine group, and nearly all of their PA counterparts navigated up the other stream. Might a salmon also use vision to aid navigation? A Japanese research team sought to find out in a study involving ocean release and recapture of sockeye salmons. The scientists blinded a sample of the fishes before release by injecting their eyes with carbon toner and corn oil. Upon recapture 5 days later, only 25% of these salmons, compared to 40% of the unaltered fishes, were caught in their natal stream. The authors suggested that these fishes are nevertheless using vision to reach the entrance to their natal stream, but I find this result unconvincing. I suspect that the pain, distress, and ensuing disorientation caused by blinding salmon with an injection of foreign substances might explain their lower success rate in finding their way home. To better control for that, they would need to have injected some salmons with a similar amount of solution that doesn't cause blindness. But I'm not recommending it. Pressure sensors. Not only do fishes navigate independently, they have another system of orientation that allows them to track closely the movements of their neighbors. Like flocking birds, who use vision and hair-trigger reflexes to coordinate their flight directions with their neighbors, large schools of fishes can change direction seemingly as one, as if there were some internal knowledge of the decision-making of all others. It isn't clear who starts it, or if the chain reaction starts with whoever happens to make the first move. Early naturalists ascribed this behavior to a form of telepathy, but analysis of slow-motion filmed sequences yields a no-frills explanation. Minuscule delays in the propagation of movement through the school show that the fishes are reacting to each other's movements. Their sensory systems are operating on such a fine timescale that it gives the impression that they all change direction as one. In daytime conditions, sharp eyesight helps schooling fishes move in unison as birds do. But unlike birds, they continue to move as one even in darkness. How? It's thanks to a row of specialized scales running horizontally along their flanks, forming what is called the lateral line. The lateral line is usually visible as a thin, dark line because each scale has a depression that casts a shadow. The depression is populated by neuromasts, clusters of sensory cells each with a hair-like projection encased in a tiny cup of gel. Changes in water pressure and turbulence, including waves from the fish's own movement reflected back from its surroundings, cause deflections of the neuromast hairs, which trigger nerve impulses to the fish's brain. So the lateral line acts as a sonar-like system and is especially useful at night and in murky waters. With the lateral line, fishes swimming in close proximity are virtually in physical contact, and the transmission of signals between them is comparable to that of visual information, giving rise to hydrodynamic imaging. It is hydrodynamic imaging that allows blind cavefishes to detect stationary objects such as rocks and coral by the distortion of the normally symmetrical field flow that surrounds a fish in open water. Blind cavefishes can form mental maps, a skill very useful for navigation by a creature lacking the means for visual orientation. Lateralization of brain functions is now known to be widespread in fishes, and these clever little fishes also use their lateral lines non-symmetrically when confronted with unfamiliar objects. When a plastic landmark was placed in their tank along the middle of one wall, blind cavefishes preferentially swam past it using the lateral line on their right side. This preference disappeared in a few hours as the fishes became familiar, and therefore comfortable, with the new landmark. Because visual and lateral line sensory systems operate independently in fishes, this finding suggests that lateralization of the brain is a deep-seated phenomenon. Sighted fishes were already known for a tendency to have a right-eye bias in emotional contexts, such as examining a new object. Like most biological designs, the lateral line comes with inevitable compromises. Water flow generated by swimming activates the neuromasts, and this background noise dampens the fish's reactivity to external movements. Experiments show that swimming fishes are only half as likely as stationary fishes to respond to the movements of a nearby predator. On the other hand, a fish can detect distortions in the bow wave formed in front of his own nose when swimming forward, and thereby avoid bumping into objects made invisible by darkness or transparency, such as an aquarium wall. It is unfortunate for fishes that this system seems unfit for detecting the presence of a fishing net. Electrified. Having a sense that allows you to avoid bumping into a wall in the dark is useful, but imagine being able to detect the presence of something on the other side of the wall when you cannot see or hear anything. Enter the world of electroreception. Electroreception is the biological ability to perceive natural electrical stimuli. It is nearly unique to fishes, the only known exceptions being monotremes, cockroaches, and bees. Electrical sensitivity is widespread in sharks, skates, and rays. Among the teleosts, more than 300 species get a charge out of life, and it must have high value as a survival tool, for it has evolved independently at least to mesenfishes. Its predominance in aquatic habitats relates to water's strong electrical conductance properties as compared to air.as. The term implies, electroreception is a passive use of electrical information. The elasmobranchs are electroreceptive only. They can detect electric stimuli, but they do not produce electricity themselves. They perceive it with a network of jelly-filled pores scattered strategically over the head. These pores are called the ampullae of Lorenzini, after Stefano Lorenzini, the Italian physician who first described them in 1678. Noting the concentration of black specks that surround shark snouts like a 5 o'clock shadow, Lorenzini peeled away the skin to reveal tubular channels, some as wide as strands of spaghetti, leading to the brain, where they congregate in several large masses of clear jelly. The function of the ampullae of Lorenzini in electroreception remained a mystery until 1960. They detect subtle electrical changes generated by nerve impulses of other organisms, which propagate efficiently through water. Such is the sensitivity of this system that just the heartbeat of a fish hiding 6 inches under the sand may be enough to betray its presence to a hungry shark or catfish. Some bony fishes actively produce their own electrical charges. You have no doubt heard of electric eels. These South American river dwellers can grow to 7 feet and 45 pounds. They are named for their elongated shape and are not true eels, belonging instead to the knifefish family, close relatives of the catfishes. They use low-voltage discharges to help them navigate in their murky habitats by detecting the electric fields that bounce off solid objects. But they are better known for producing stunning electrical discharges up to 600 volts or more. The electric organs are housed in stacked cells within the tail musculature. As in the stacked cells of a battery, electricity can be stockpiled until needed, then, if the eel so chooses, released all at once. This built-in taser gun can be used to stun or kill prey, or to repel unwelcome intruders. The voltage power of electrical discharges of electric eels and some other fishes, such as torpedo rays, have earned them the term strongly electric fishes. But for me, the most interesting use of electricity is reserved for certain weakly electric fishes, who use it for the less violent purpose of communicating with others of their kind. Most of these fishes belong to two groups: the diverse elephant fishes of Africa, so named for their elongated, downward-pointing noses, and the knife fishes of South America, named for their pale coloration and knife-like shape. Like so many fishes with stealth technologies, they inhabit muddy waters, which likely provided the adaptive basis for a novel non-visual means of communication. They communicate with high-speed electric organ discharges of up to 1,000 pulses per second, or 1 kHz, more than twice as fast as an electric eel's pulse rate. They are adept at interpreting these signals, as illustrated by a species of elephant fish that lives in river and coastal basins of Western and Central Africa. When the biologists Stefan Paintner and Bernd Kramer from the Institute for Zoology at Regensburg University, Germany, presented them with simulated eodes, the fishes showed an astounding ability to discern pulse time differences down to a millionth of a second. This rivals echolocation by bats as the fastest form of communication in the animal kingdom. By varying the rate, duration, amplitude, and frequency of their eodes, elephant fishes can exchange information about species, sex, size, age, location, distance, and sexual inclination. Eodes also communicate social status and emotion, including aggression, submission, and mate attraction, for which signals are crafted into courtship songs to serenade potential mates using exotic patterns of chirps, rasps, or creaks. They can identify other individuals by their eode signatures, which are distinct and remain stable over time. Dominant individuals may chase trespassers off their territories when they detect the trespasser's eode, which probably explains why fishes often deferentially turn off their eode when swimming through a neighbor's territory. Pairs or groups of fishes also coordinate their eodes, producing echoes and duets. Males will alternate eode pulses with other males, whereas females will synchronize theirs with investigating males. It could get confusing when a cluster of elephant fishes or knife fishes are chirping away in close proximity. They deal with it using a so-called jamming avoidance response. If two fishes' discharge frequencies are too similar and might interfere with discrimination, they adjust to enlarge the distinction. Fishes in a social group maintain a 10 to 15 Hz difference from neighbors, ensuring that each individual has a personalized discharge frequency. Recordings of eode-producing elephant fishes in the upper Zambezi River suggest that they also use their signals to cooperate. Eodes produced by fishes threatened by a lurking predator induce neighbors to join in on what may be an early warning alert. It benefits all fishes in the neighborhood if predators have low hunting success there. Signals exchanged by familiar neighbors can provide assurances that all is well, thereby avoiding the need for costly defense of territory. Such dear enemies also team up as shoaling partners when food gets scarce. If all this sounds too sophisticated for a fish, it may be time to reassess your perceptions of fish intelligence. Consider also that the elephant fishes have the largest brain cerebellum of any fish, and that their brain-to-body weight ratio, a highly touted marker of intelligence, is about the same as ours. Much of that gray matter is devoted to electroreception and communication. There is a cost to using electricity to communicate. Electroreceptive predators could be tuning in. Such is the case with sharp-toothed catfishes, who hunt in packs during spectacular yearly migration runs up the Okavango River in southern Africa. Most of their diet during this time is a species of elephant fish called the bulldog. They locate the hapless bulldogs by eavesdropping on the bulldog's eodes. But there's a further twist. Captive studies have found that the eodes of female bulldogs are too short for the catfishes to detect, whereas the male's eodes are 10 times longer, and the catfishes can easily notice them. The size distribution of bulldogs found in catfish stomachs indicates that it is the males who are mostly being eaten. In the evolutionary arms race of avoiding being someone else's meal, we may expect male bulldogs to be shortening their eodes. The pleasure of touch. While lateral lines and electric organ discharges are alien to our sensory systems, the sense of touch certainly is not. In exploring this familiar sensation in fishes, I want to connect it to another kind of sensation that we often derive from touch, and one that we rarely consider as being part of the lives of fishes. I am referring to the sense of pleasure in his iconic poem Fish, D.H. Lawrence wrote. They drive in shoals, but soundless and out of contact. They exchange no word, no spasm, not even anger. Not one touch. Many suspended together, forever apart. Each one alone with the waters, upon one wave with the rest. I love these lines, and I can see what Lawrence means. To my airborne senses, there is something lonely about fishes being forever suspended in their heavier, viscous medium. But writing in the early 1920s, Lawrence didn't have the benefit of knowing what we know today about the lives of fishes. Fishes are not alone. They know each other as individuals, and they have preferences for who they hang out with. They communicate through diverse sensory channels. They have sex lives. Contrary to the notion of their being separate, it turns out that fishes are highly sensitive to touch, and tactile communication enriches the lives of many. While researching this book, I was sent a video clip by a puzzled viewer who couldn't understand why a fish, in this case a bright orange Midas cichlid who looks for all the world like a friendly character from Finding Nemo, would return repeatedly to be stroked, picked up, and playfully tossed back into the water by a man. What would motivate a fish to do this? The answer, I believe, is that it feels good. Fishes often touch one another in pleasurable contexts. Many court with rubbing or gentle nips. Cleaner fishes curry favor with their valued clients by caressing them with their fins as a means to strengthen the cleaner-client relationship. Moray eels and groupers approach familiar divers and receive strokes and chin rubs. In an informal survey of public perceptions of fishes, I received unsolicited accounts from 8 of 1,000 random respondents who described behavior like that of the Midas cichlid we just met. These fishes would allow their humans to pet, touch, hold, and stroke them. The author Kathy Unruh later wrote to me about a Bahamian grouper she calls Larry. Whenever Kathy and other divers descend to his reef, Larry swims over to be petted. According to Kathy, Larry seems to enjoy making eye contact and checking out the diver's bubbles. He even rolls side to side to be petted properly, as a dog or a pig will do. Today one can find videos of fishes cavorting and in some cases appearing to snuggle with divers, who stroke their bodies gently as if they were the family cat. There are also growing numbers of videos of aquarium fishes swimming repeatedly into the hand of a trusted owner to be lovingly stroked. The other major group of fishes, sharks, rays, and skates, also show pleasurable responses to touch. The diver Sean Payne described an encounter he had with a juvenile manta ray off the Florida coast. The ray swam up to Payne and rubbed repeatedly against him, leading him in a circular tango that forced her body into his hands. As I ran my hands over her skin, her wingtips vibrated like a dog's leg during a particularly good belly scratch, said Payne. Andrea Marshall, the founder of the Marine Megafauna Association, describes manta rays as strongly curious and interactive with humans. These massive elasmobranchs, who have the largest brains of all fishes, love getting bubble massages from Marshall. She swims beneath them and blows bubbles from her scuba regulator. If she stops, the rays swim away, but soon return for more. It's a similar story at the Shedd Aquarium in Chicago, where 2 of the 5 zebra sharks in a 400,000-gallon tank like to swim among the staff divers. I think they like the feel of the bubbles coming out of our regulators, says Lise Watson, Wild Reef Collection Manager. During our maintenance dives, if we put our regulators underneath them, they dance around while the bubbles tickle their bellies. Besides touch, there are many other ways that fishes may derive pleasure. Food, play, and sex spring to mind. And then there's comfort for its own sake. Southern bluefin tunas in the waters of Australia spend hours rolling on their sides, catching the sun's rays. It's not known for sure why they do this. One possibility is that they are sunbathing to raise their body temperature, which in turn helps them swim and react faster, making them more efficient hunters. I expect the warmth of the sun also feels good to a tuna, for pleasure evolved to reward useful behaviors. Ocean sunfishes are renamed for their fondness for sunbathing while lying on their sides just beneath the surface. These huge fishes are also parasite hotels, harboring as many as 40 different species of external parasites, including large copepods that can reach 6 inches. The sunfishes queue beneath floating kelp beds, waiting their turn to be serviced by cleaner fishes there. The sunfish at the front floats onto its side to signal readiness. But some of the parasites are too large to be removed by fishes, and this is when the sunfish turns to a specialist. Floating up to the surface, the giant fish invites gulls to surgically remove penetrating skin parasites with their powerful beaks. Sunfishes have been seen courting the birds, following them around and swimming sideways next to them. Dare we think the sunfish knows the feeling of relief from a skin irritation and understands the cause and effect of bird and parasite? It is the best fit explanation I can think of for a wise old creature who may live a century and wander thousands of square miles of open ocean. To know pleasure is to know pain. Or so it would seem. Yet, despite steady advances in our understanding of the full-bodied lives of fishes, the question of their capacity to feel pain remains a subject of debate. Should it? Let's find out. Part 3. What a fish feels. Your life a sluice of sensation along your sides. From Fish, by D. H. Lawrence. Pain, consciousness, and awareness. Water wetly on fire in the grates of your gills. From Fish, by D. H. Lawrence. Do fishes feel pain? While it may seem obvious to some of us that they do based on their appearance, their behavior, and their membership in the group of vertebrate animals, many people believe otherwise. I am only aware of limited opinion research on this question, such as a survey of North American anglers and other recreational fisheries stakeholders, which found that slightly more believed fishes feel pain than believed they do not, and a survey of New Zealanders that had a similar result. The question of whether fishes experience pain is of cardinal importance. Recall those astronomical numbers of fishes killed by humans from the prologue. Organisms that can feel pain can suffer, and therefore have an interest in avoiding pain and suffering. Being able to feel pain is not a trifling thing. It requires conscious experience. An organism may move away from a negative stimulus without any experience of pain. It could be a reflexive response in which nerves and muscles cause the body to move without any mental engagement. For example, a heavily sedated human patient in a hospital setting with no capacity to experience pain may nevertheless recoil in response to a potentially harmful stimulus, such as exposure to heat or intense pressure. This is due to the actions of peripheral nerves working independently of the brain. Scientists use the term nociception to describe a reflex that in itself involves no awareness or pain. Nociception is the first stage in pain sensing, necessary but not sufficient for the experience of pain. It is only when information from nociceptors is relayed to higher brain centers that it hurts. There are some good reasons to expect that fishes are sentient. As vertebrates, they have the same basic body plan as mammals, including a backbone, a suite of senses, and a peripheral nervous system governed by a brain. Being able to detect and learn to avoid harmful events is also useful to a fish. Pain alerts animals to potential damage that may lead to impairment or loss of life. Injury or death reduces or eliminates an individual's reproductive potential, which is why natural selection favors the avoidance of these dire outcomes. Pain teaches and motivates animals to avoid a noxious past event. I have an assignment for you that might provide some insight on the question of whether fishes are consciously aware and thus capable of pain. Go to a public aquarium. Choose a tank. Spend 5 minutes watching the fishes in there. Look long and hard. Look closely at their eyes. Watch the movements of their fins and their bodies, keeping in mind what you now know about their vision, hearing, smell, and touch. Choose an individual. Does he pay attention to other fishes? Do you see any organization to his movements, or does he appear to be just randomly swimming about as if on autopilot? If you do this, you will usually see non-random patterns of behavior. You'll notice a tendency for fishes to consort with others of their own kind. You will see, especially in larger fishes with more easily watched body parts, that their eyes are not locked into a fixed stare, but swivel in their sockets. If you are especially patient and observant, you'll note idiosyncrasies expressed by individuals. For example, one fish might appear dominant over another, giving chase when the subordinate transgresses some social or physical boundary. Some individuals may be more adventurous, others more shy. When I was little, I didn't pay much attention when gazing at fish in a tank. I wasn't looking at other beings, only at swimming creatures with shapes and colors. Gradually, I began to watch fishes more closely, and they became